Good afternoon and welcome to the Department of Biochemistry at the University of Cambridge in the UK. My name is Dr. Rhys Grant and I am the Science Communications Specialist here in the Biochemistry Department, shared with the Cell and Molecular Biology programme at the Cancer Research UK Cambridge Centre at Cambridge University. So I'm very pleased to welcome you to our department today for this first live talk as part of the brand new 2021 Cambridge Festival, which aims to bring the research of the University of Cambridge direct to you in your living room, office, garden, or wherever it is that you're watching us from this afternoon. So for this first talk, I'm joined today by Professor Sarah Lummis, who is Professor of Molecular Neurobiology at the University of Cambridge. And today, Sarah is going to be speaking to us about the molecules that cells use to communicate with one another. So how these molecules evolved, why these molecules are important, and how understanding the biochemical pathways that these signaling molecules are involved in can help explain to us the actions of some of the most widely prescribed drugs, such as Viagra and Valium. I'm also joined today by Latika Sugamba, who is part of our department's secretariat team, and who's going to be helping me out behind the scenes with running this live event. Before all of that though, the first thing I want to ask you all to please do is click the subscribe button down below to follow our YouTube channel, because we have loads more talks coming up this week as part of the Cambridge Festival, and we're planning on bringing you even more live content from our department in the near future. You should also go and check out our other social media channels on Twitter, Facebook, Instagram, and so on, using our handle, which is @CamBiochem. So that's CAMBIOCHEM, because we use these channels to bring you all of the news, research, events, and information about everything really that's going on in our department. I also just want to mention that after today's talk, we'll be having a live question and answer session with Sarah and myself. So if you have any questions on Sarah's talk, or perhaps if you'd like to ask us something more general about maybe what life is like as a life scientist, please just post your questions in the live chat on YouTube, and then Sarah and I will stay for about 15 to 20 minutes after the talk to answer as many of your questions as we can get through. If you do participate in the Q&A session, you don't need to worry about having a lockdown haircut or anything like that, because you won't be seen and you won't be heard. It's just going to be us reading your questions out anonymously on your behalf. So as I mentioned, today's talk is going to be given by Professor Sarah Lummis, who is Professor of Molecular Neurobiology at the University of Cambridge in the UK. So Sarah has a particular interest in the molecules that are involved in communication or signaling between different cells. And interestingly, Sarah's favorite molecule can be found in what's known as the vomiting center of the brain. So as you might guess from that name, when this protein is activated, it will unfortunately make you feel sick. But despite that, Sarah has spent many happy years researching this protein and its relatives, which has led to her publishing over 100 primary publications in the literature. In addition to Sarah's research and teaching roles that she does in our department, Sarah is also a fellow and director of studies for natural sciences at King's College in Cambridge. And she's hoping that she's been sending her students all the right signals for being productive, disciplined, and intellectually curious in the life sciences. So with that, I'm going to hand over to Sarah for this afternoon's talk. Thanks, Sarah.
Well, thank you very much, Rhys, for that introduction. And welcome to this talk. As Rhys said, I am Professor Sarah Lummis. I'm based in the Department of Biochemistry, but I'm also associated with King's College. And here on my introductory slide, I've actually got a picture of my favourite protein that Rhys just mentioned, and I'll come back to that briefly a little bit later on. But just to give you a clue, I have on the right-hand side a rather bad drawing that I did of a nerve cell or a neuron, and this is where my protein is usually located. And again, I'll come back to that a little bit further on in the talk. Okay, so what is the aim of today's talk? Well, I want to try and give you a little bit of a feeling of what the knowledge of signaling between cells can do for us, and also perhaps give you some of the excitement in actually studying this area. And I'm going to do that by sort of asking a question. So is this signaling an ancient phenomena, or is it actually just something that's really very modern? And I'm going to illustrate that with two different signals. One that I'm calling here an ancient signal, and this is nitric oxide. Now, I'm very sorry, we have lots of acronyms in this field, so we tend to talk about this as NO. But I'm also going to talk about a modern signal. So that's this molecule here, gamma aminobutyric acid, which we refer to as GABA. Okay, so before I get started, I just wanted to give you a bit of an introduction to why do we need any signals between our cells? And what I have up on this slide, on the right-hand side, is an image, well, a cartoon of two cells. So we have a green cell up here, a grey cell on the bottom. Now, of course, cells we could probably consider as a sort of smallest units of life, and often referred to as the building blocks of life. And all they are really a mass of cytoplasm enclosed within a membrane, which contains many biomolecules such as proteins and nucleic acids, and they allow the cell to function. So getting back to why signaling is important, the first reason I have up here is that organisms need to be able to interact with the environment. And we know from our own experience how important it is that we can, for example, see and hear our environment. So you didn't trip over your computer cables when you were rushing to your computer to actually switch on this talk, for example. But what you may not be aware is that all living things are continuously bombarded by signals in many different forms. And it's the ability of multicellular organisms or individual cells within an organism to sense and respond to their environment that's crucial to their survival. Now, to do this, cells need the ability to detect the presence of extracellular molecules and conditions, and then instigate an appropriate response. And what we can see here on our bottom cell is a whole range of signals. So we get sensory signals, we get nutrient signals, and we get signals from other cells. And there are a few things also happening inside the cell that are in response to these particular signals. So this brings us to the second point on our list here, that signaling is needed to convey information. So information, as I mentioned, comes in a large variety of forms. So a signal from a neighboring cell, for example, the green cell up here, often produces some sort of molecule that's then detected or received by the target cell, the bottom one. So single cells are complex enough, but obviously there's a whole another dimension of complexity when multicellular organisms are considered. So here the cells that need to communicate may be very distant from one another, and they might have very specific and very different functions. And that brings us to our third point here, that signaling is needed to facilitate complex coordination. So let's move on now to a specific example in the next slide. So here what I've got is a developing embryo. So we start with a single cell, a fertilized egg, and then this divides, eventually reaching a blastocyst and then a full organism. Now, each cell here at every stage has to communicate with all the others, such that each gets a specific role in the final organism. And in fact, these signals determine whether an embryonic cell will survive, will divide, or in fact die. And it's perhaps worth keeping in mind that most cells that do not receive signals are actually programmed to die. Okay, so that's a little bit of an introduction to cell signaling. So let's get back to my two examples that I'm going to be talking about. Okay, so I've said here that nitric oxide is my example for an ancient signal. Well, how ancient do I actually mean here? If I tell you that nitric oxide is actually a common constituent of star-forming gases throughout the cosmos, you might realize that we're going back an awfully long way. And in fact, let's go back to the formation of the Earth. So here I've got a little time plan here. So the formation of the Earth was about 4.6 billion years ago. And at that time, a lot of nitric oxide was actually being formed. And this was by chemical reactions involving nitrogen and carbon dioxide, which were plentiful in the Earth's atmosphere at that particular time. So this was in fact driven by electrical discharges, because there were a lot of those that happened during this time. Okay, I keep talking about this time. It's actually known as the Hadean era. And Hadean is after Hades, the god of the underworld who guarded the entrance to hell. And that's because there was supposed to be very hellish conditions on Earth. The Earth was very hot. There were lots of radioactive elements. There was bombardment from other solar system bodies. And there was lots of lightning. And I had a little image here that you might have seen that sort of, I thought, gave me some idea of what it would be like to be on the Hadean Earth. And I'm not sure if you can see that. This says it's the Earth, Jim, but not as we know it. So apologies to Star Trek fans, or perhaps the non-Star Trek fans, for adding that little bit in there. Okay, so the Hadean Earth, not a very pleasant place to be, but there was lots of nitric oxide. So let's go back to our little plan here. Okay, so why is nitric oxide important here, and what was it doing? Well, interestingly, what nitric oxide was the intermediate that linked our ability, that the nitrogen that was present already in the atmosphere, but was in a very stable form and couldn't be used, to the nitrogenous substrates that were actually needed for the building blocks of life, for things like purines, pyrimidines, and amino acids that we need for protein and for DNA. So the nitric oxide allowed the formation of these particular molecules. Okay, so once these were actually available, then the stage was set for the evolution of terrestrial life forms. And we can see up here, so this is approximately when prokaryotes evolved sort of towards the end of this Hadean era. Okay, so nitric oxide is perhaps in effect telling us that there's sufficient nitrogen to actually sustain life. And nitric oxide provided the necessary source of this fixed nitrogen to fuel life through most of the Archean period, which you can see is the next period in the Earth's history. But atmospheric simulation experiments suggest that a crisis in the biosphere occurred towards the end of this period, when the production of nitric oxide is estimated to have waned about 100-fold. And that was apparently due to diminishing CO2 levels. Now, this is proposed to have caused such a severe ecological crisis that it provided the selective pressure for organisms to evolve the ability to perform nitrogen fixation, and also the enzymes which actually could synthesize nitric oxide evolved. So if we just move on here. Oops. Okay, so we were still getting nitric oxide towards the end of the Archean period. Now, by this time, you know, the prokaryotes, the bacteria that we had evolved quite a long way. They were making enzymes for use in metabolic pathways. And that was great. But there was a bit of a problem, because nitric oxide is actually toxic to the catalytic activity of transition metal centers that are present in a number of metabolic enzymes. So, so what was going on? Now, about this time, ancient hemoglobins arose in bacteria. Now, I'm sure you've all heard of hemoglobins, and they bind oxygen. But in fact, they bind nitric oxide rather better than they actually bind oxygen. So the hypothesis is that these actually arose as a biodefense mechanism, first for nitric oxide, and then when oxygen had accumulated significantly on Earth, which was due to the emergence of photosynthetic bacteria, then they were used to scavenge oxygen, which also was toxic to these particular enzymes. So just to remind you of hemoglobin, here we go. I'm sure this looks familiar to many people. So here's our red blood cells, and they are full of this protein, hemoglobin. And hemoglobin contains a number of heme groups, and it's these heme groups which contain iron that actually will bind oxygen. And I think we have, for those more interested in the structure, we have a little chemical image here of our heme group, and this is where our nitric oxide actually binds. Okay, so I mentioned that the enzymes to make nitric oxide evolved towards the end of the Archean period. Okay, but are they still here today? And the answer is yes, very much so. In fact, we not only have one enzyme as a mammal, but in fact we have three. And this is because nitric oxide, is still an important signal today. So these enzymes up here, NO-producing enzymes, which are called NO synthases or NOSes, as I've said, they emerged many eons ago, but the higher eukaryotes, such as humans, have evolved three distinct forms of these. Okay, so what does nitric oxide do? Well, it's actually a very important signal in the nervous system, so present, for example, in the brain, but also in the immune system, which we all know a lot more about now than we did a year ago, and in the circulatory system. So I thought I'd give you a little bit of an example of what this does. Okay. So one site of action of our nitric oxide is in endothelial cells. So these are just flattened cells that, amongst other things, line every blood vessel in the body. And you can see them here in yellow. Now, these endothelial cells release nitric oxide in response to nerve stimulation. Here's our nerve coming in on the left-hand side. Now, the nitric oxide causes the smooth muscle cells that line the vessel wall, and you can see them just behind the endothelial cells, to relax. So that allows the vessel to dilate, so blood flows through it more freely. Now, the effect of nitric oxide on blood vessels accounts for the action of nitroglycerin, which is also called amyl nitrate, which has been used for hundreds of years to treat patients with angina. Now, angina is just pain caused by inadequate blood flow to heart muscle. Now, in the body, nitroglycerin is converted to nitric oxide, which rapidly relaxes blood vessels, thereby reducing the workload on the heart and decreasing the muscle action needed to produce oxygen-rich blood. Okay, so that's our nitric oxide effect. So I want to have a little look at the biochemical pathways. I'm a biochemist, so I'm very interested in these. So we know quite a lot about this. And what you can see in this image is our nitric oxide being synthesized in our endothelial cell, as I mentioned. And here's our enzyme, nitric oxide synthase. And it's basically a reaction involving arginine, which is a very important amino acid, which interestingly contains a lot of nitrogen. Okay, so the nitric oxide that's produced then diffuses to the neighboring smooth muscle cell, and it binds to another enzyme. So this is guanylate cyclase. And what does this enzyme do? It actually converts this molecule here, GTP, to cyclic GMP. And this is what causes the relaxation of the smooth muscle cells. Okay, so I want to take a closer look at the guanylate cyclase for a moment. So here it is. Here's a rather bad diagram of a guanylate cyclase enzyme, which is a dimer. So it has two subunits, and they're called alpha and beta here in yellow and green. And what you can see is that it also possesses a heme group, and that's what this is up here with our iron in the middle. Now, what's particularly interesting about this is that the nitric oxide binds to the heme group in exactly the same way as it does in hemoglobin. And in doing so, it actually changes the shape of this heme molecule. And I've shown that down here. So the heme molecule is normally domed. And once the nitric oxide binds, it actually is planar. So what that does is pull this particular residue, this amino acid, this is a histidine molecule, towards the heme group. And so now we have a conformational change. The enzyme changes shape and becomes active. and we start making our cyclic GMP. So that's really interesting, and we know what's going on here. And I can show you a little image up here of the actual structure. And here is our protein, and we can see the heme group right in the middle here in green, and it's nice and flat. So this must be an active enzyme. For those of you that know these things, there's our histidine molecule that I was talking about before, underneath our heme molecule. Okay, so why are people interested in nitric oxide? Obviously, it's important, but one reason is because it appears to play a role in anaphylaxis. Okay, so you'll all be aware that anaphylaxis is a sudden and severe systemic allergic reaction, and it can be caused by insect bites, reactions to medications such as penicillins and anesthetics, foods, nuts, fish, shellfish, a particular problem. And those who are actually struck by anaphylaxis find their face, neck, and throat swelling up. They have trouble breathing. They may eventually lose consciousness as their blood pressure drops, and they can cause death if the symptoms are not treated. Now, anaphylaxis was considered to be quite rare, but it's now becoming apparent is not as rare as generally believed and may affect as much as 15% of the population. In addition, and somewhat worryingly, the prevalence of anaphylaxis is increasing significantly. Okay, so, and just to give you a little example here, you may remember this from a few years ago. There was a 15-year-old girl who was severely allergic to sesame seeds and died after eating a sandwich that did not list the ingredients on its packaging. Okay, so she bought this at an airport, she got on a plane, she had this allergic reaction on the plane, and unfortunately, she collapsed and died actually on the flight. So, as I have said, during an anaphylactic reaction, acute hypertension is the most important clinical feature. But to date, supportive adrenaline and large volumes of intravenous fluid resuscitation are really the only available treatments, and not all patients respond. And sadly, this girl did not. Now, these treatments, however, don't really address the key signaling pathways involved in the process, but just address the general symptoms. So as I mentioned, a recent role of nitric oxide and anaphylactic shock was uncovered, and this was demonstrated in mice that were pre-treated with an inhibitor to the nitric oxide synthase. And this compound successfully prevented these mice from developing both the hypertension and death when they were put into anaphylactic shock. So going back to our slide, let's have a look. So this compound was actually an arginine mimic. So if we go back to our reaction pathways, what we can see is that arginine is needed to make our nitric oxide, and it actually binds to our nitric oxide synthase. But if you have a compound that mimics the arginine, it can prevent the action of our nitric oxide synthase. So really interesting, lots of work going on in that particular area. Okay, so there's another drug that again is very interesting that also works by this pathway. And if we move on to talk about the final enzyme that I haven't really mentioned yet, this PDE5, and what this does is basically it removes the cyclic GMP from the system, breaks it down to GMP. So this enzyme, PDE5, actually stands for phosphodiesterase 5. And the drug that I'm talking about that works in this system is actually Viagra, which I think everyone's probably heard of. It's to date netted Pfizer over $25 billion in worldwide sales, but it was originally developed as a cardiovascular drug. And in fact, as an inhibitor to this particular enzyme, phosphodiesterase 5, the idea being to block the enzyme, maintain high levels of cyclic GMP, leading to relaxation of smooth muscle and lowering the blood pressure, just like nitroglycerin does. Now, it turned out the Viagra was relatively useless for this, but it can prolong and enhance stimuli, but has little or no effect in the absence of stimulation. So we might, however, all be aware of the side effects that were experienced by participants, and apparently this was notably the elderly male participants, in the early trials of Viagra, when it was being tested as a cardiovascular drug, I have to say. And that has led to its prolific worldwide use, because Viagra certainly does act to enhance the effect of nitric oxide, which of course controls blood vessel dilation in erectile tissue, as well as in the heart. And I think we have a little image here. Oh, yes, of Viagra actually bound to the phosphodiesterase enzyme. Those of you that can see it, again, this is a molecule that actually mimics the cyclic GMP in binding to the enzyme. And you can see that the structure of Viagra is fairly similar to the structure of cyclic GMP. Okay, well, I think that's probably enough about nitric oxide and our ancient signals. So I want to move on now to talk about what most people would consider is a more modern signal. And this is our friend GABA. So let's have a little look. So in fact, this is really important in nerve cells. And what are they? So they're just specialized electrical signaling cells that we find in multicellular animals. So this is our most important signaling cell. So when activated by a signal, maybe from the environment or maybe from other cells, then the neuron sends electrical impulses racing along its axon at speeds of up to 100 meters per second. And when these reach the axon terminal, the electrical impulses are converted into a chemical signal called a neurotransmitter, which is released into the narrow gap that separates the axon from its target cell. So I want to look at this region, which is called the synapse, in a little bit more detail. I've got that in the next slide. Okay, so our blue here is our nerve cell. And inside this nerve cell, we have little vesicles that are filled with chemicals, chemical neurotransmitters. So when the action potential reaches this part of the cell, these vesicles release their neurotransmitter, and they then bind to special proteins in the adjacent cell, specific receptors for these neurotransmitters. And what this can do for certain proteins will then open an ion channel and allow ions to actually flow in down the channel. So I think you see in the bottom here, I've blown up two of my receptors, and this one has not got any molecules bound, and this one does. And you can see the structure is subtly different, such that we now have a gap here through which the ions can actually flow into the cell. Now, there are a whole range of different proteins that will bind neurotransmitters. The family of proteins I'm particularly interested in is called the pentameric ligand-gated ion channel family. And one of the neurotransmitters that acts on those is GABA. And I've shown the structure of GABA down here, this gamma-aminobutyric acid. So we can call these perhaps GABA receptors if this molecule is GABA, is activating these proteins. So these are very important proteins. I'll talk a bit more about them in a moment. But this surely must be a very modern signal, because obviously we don't have brains in the early Earth. So let's go back to our little plan here. Okay, and about 600 million years ago, so down here about over here on this location, the common ancestor to all animals gave rise to some organisms with simple neural networks. So presumably this is, in comparison to nitric oxide, a fairly modern signal. So then we wouldn't expect to find it in organisms that are particularly ancient, like some of our ancient bacteria. Now, it turns out that we know quite a lot now about the genomes of our bacteria, so we can identify what molecules actually are present in these bacteria. And what was particularly astonishing was that in one particular bacterium that was being studied, it turned out there was the code to make a GABA receptor that I've just been talking about. So if we look at our, so we've got a phylogenetic tree here. So we've got some sort of common ancestor back here that gave rise to our bacteria that we've been talking about, but also through evolution to all sorts of other species as well, that we now consider are probably much more highly evolved than our bacteria. So to actually find a GABA receptor in bacteria was very strange. So let me just show you a picture of these bacteria up here. So it's actually, so here are the bacteria, they're species called Owinii, and they're actually very well known because they're very damaging to crops and particularly potatoes. So I believe they're the cause of the Irish potato famine hundreds of years ago, and you can see what they actually do to potatoes. They rot the potatoes. So again, so we know all about the genome of this, and this code was identified. So that was a challenge. You know, did it actually look like a GABA receptor and behave like a GABA receptor? And the answer, somewhat surprisingly, is yes. So let me show you its structure. So we're coming back here to my favorite protein on the right, just as a comparison. But here is the protein that I've been talking about. And it's actually been named ELIC, had to be named something with an acronym. And that stands for Owinii ligand gated ion channel. And what you can see from these structures, that it's very similar to this GABA receptor that I was talking about earlier. And that's also very similar to my favorite protein, which happens to be the 5-HT3 receptor, which had to get a little look in somewhere. Okay, so does that mean that in fact, GABA was an ancient signal for bacteria? And I wanted to show you that actually chemicals, small chemicals, can be really important for signals in unicellular organisms. So I'm hoping I can show you this little movie. Now, this isn't actually a bacteria, it's actually an amoeba, but the principle is the same. So here's our little amoeba. And what you may be able to see, I hope you can see here, is a pipette that's actually releasing a chemical. Now, this chemical could be a food source for the amoeba, or it could just represent that there is food, so a signal that's released from food, so that the amoeba knows which direction to go. And if I play this movie, I think you'll be able to see that this pipette and its chemical has a very dramatic effect on where this amoeba actually goes. And I just say, you know, no amoebae were harmed in the making of this particular movie. Okay, so chemicals are really powerful signals, not just in your body. I'm sure everyone's aware of sort of hormone effects, but also to single-celled organisms. Okay, so that was our amoebae. Going back to the effect of GABA on our Owinii bacteria, did they actually have any effect? So I thought I'd just show you a bit of data here. No, we've finished with you. Okay, so here it is. So I've said we know a lot about the genome of those Owinii. There's some very clever people in the salmon group in the biochemistry department who can take genes out of these bacteria. And if we take the gene out of ELIC, and this is actually data from a colleague of mine, Hoya in France, that it has a dramatic effect on the effect of GABA. So here in our normal bacteria, if we apply GABA, these bacteria will actually move, but if you take the gene out, then there's practically no movement. So it seems that GABA is actually also an ancient signal that was found in bacteria. And perhaps here it was actually signaling the most important thing to these organisms, which was the presence of something to eat, because GABA is present in many organisms, and in fact was identified in potatoes before it was identified in humans. And perhaps later, these molecules became adapted for internal communication. So internal communication, what do these GABA receptors do in you and I, in mammals? So I just want to spend a couple of minutes talking about that. So this is going back to the GABA receptor that I was talking about earlier. And our little blue boxes here represent the binding sites for the neurotransmitter GABA, which will activate this protein. But you can see there are all sorts of other places that colors that I've decorated, which tell you that there are other molecules that will also activate this receptor. Perhaps one that's really interesting is that for benzodiazepines. So these are drugs such as Valium, Rohypnol, sort of really commonly used drugs in the Western world. And they, in combination with GABA, will actually activate GABA receptors in the brain. We've also got things like anesthetics, we have steroids, and alcohol will all modify the effects of GABA. So understanding what these do is really important and also really useful for the development of new drugs. So that's really important. But also, as you might imagine, malfunction of these proteins causes a whole range of neurological disorders. And one that I just want to talk about for a few moments is epilepsy. So this is a really common problem. So 1 in 26 people will develop epilepsy in their lifetime. So that makes it the 4th most common neurological condition, affecting more than 65 million people worldwide. Now, epilepsy can develop in any person at any age, but it's more common in young children and in older people. And many famous people have actually suffered from epilepsy. So we've got a few people down here. So Lewis Carroll, and apparently falling down a rabbit hole is a familiar feeling to people with seizures. Julius Caesar apparently fell convulsing into the river Tiber. Vincent van Gogh was supposed to have some of his most productive periods when he was just pre-epileptic. And this is the musician Prince again. Some people consider him a genius. Again, he suffered from epilepsy. And what we know is that mutations in the GABA receptor can be directly responsible for some of these epilepsies. And I've put some of them on the next slide. So these little red images here just shows where we know there are mutations in the amino acids that are directly linked to a particular type of epilepsy. And there are lots of different types of epilepsy. So up here we have some related to childhood absence epilepsy. So this is a really interesting epilepsy. So typical story, let's take a child, Sonia, who's 8, who blanks out and stops dancing for 10 to 20 seconds during her ballet lessons. Then she's right back where she started in her dance routine. So the seizures, sort of staring spells of about 10 seconds, and the child isn't aware or responsive. And they can occur up to 50 times a day. So very disconcerting. Although interesting, the children will often grow out of these seizures. Now, I've put a couple of other types of epilepsy down here, and these are generally more serious and quite often they're fatal. Okay, so I think my time is coming to an end. So what have I been telling you about? Okay, the signaling. So hopefully you've come to some ideas of whether it is an ancient or a modern phenomenon. And I talked about nitric oxide, which I defined as an ancient signal, but I think you now know it's also a modern signal. And I talked about GABA, which I said was a modern signal, but we're also now getting evidence that it's an ancient signal. So it looks like this signaling has been around for many years. I hope it'll be around for a few more years to come, because I'm really enjoying doing research and understanding the systems. And it just leaves for me to say, thank you for all the people that have worked with me over the years. I've had some fantastic students, postdocs, collaborators. Been very fortunate to be funded by lots of wonderful people. And I think now I'll just hand back to Reese, who's going to move us on to the next stage.
Oh, thank you very much, Sarah. That was genuinely fascinating. A real grand tour through so many different aspects of cell signaling. Really interesting. And so we're now going to have our live question and answer session. So if you have any questions, please just post them in the live chat on YouTube, and then Sarah and I will answer as many of these as we can get through. So because there is a slight delay, I'm going to go first with a question that I've got. Sarah, you mentioned nitroglycerin to treat angina. Nitroglycerin is an explosive.
Yes, you've got to be a bit careful of the levels that you use. Yes. So yeah.
So there's no historical cases of people accidentally exploding from their angina treatments?
Not that I'm aware of. That's an interesting question. Yeah, maybe hundreds of years ago they did, but I think you have to have it in the right proportions with other things to make an explosive. I don't think it explodes on its own. So.
Okay. And I was wondering also if you could tell us a bit about your favorite protein and the vomiting center in the brain.
Well, actually, yeah, it's really interesting because it wasn't thought that this protein was responsible for vomiting. It just so happened that the most, probably 30, 40 years ago, the most useful drug happened to act better on a different protein than on my protein. However, once my protein had been discovered, it became apparent that actually this is the one we should be looking at. And the drugs that now you get offered for any sort of nausea and vomiting, and anyone who has will probably be given something like ondansetron, that will completely block my favorite protein. So, and we didn't know that until we started working on it. I was very fortunate to be in right at the beginning. Actually, it was with Glaxo, started a collaboration on this protein, and they were the people that developed ondansetron that we showed blocked this receptor and blocked nausea and vomiting. And that, I think, is one of the biggest problems of people undergoing chemotherapy, that they just hate the nausea and vomiting associated with the treatment. And this really wiped that out. So it's actually, even though it sounds like a fairly minor ailment, it's actually really important to be able to inhibit that so that other things can be used.
Is that the same with seasickness and things? Would that be an inhibitor against, was it 5-HT?
Yeah, no, that's a slightly different pathway. Okay. So it hasn't actually been used for that, although, people are now starting to use it for other nausea and vomiting tricks that we never thought it would work for, but it does seem to be having some effect sometimes.
I'm not seeing any questions yet on the YouTube chat, so I'm going to ask another one. Because you're the Director of Studies for Natural Sciences at King's College, I was wondering if you could share a little bit of information about what the undergraduate course in natural sciences is like at Cambridge, because I'm sure there'll be some people watching this who aspire to be students here in Cambridge in the future.
Well, it's a fantastic course. I mean, okay, I'm slightly biased at that. But one of the major advantages is that you have so much choice in which direction you want to go. So as long as you've got some scientific background, that you can choose between a whole range of different subjects in your first year, and again, and even more in your second year. So you might end up doing something that might be completely not what you thought you were interested in, but because you'd never met it, you didn't realize that actually that was a particularly fascinating subject. So I've sort of jumped between biochemistry, pharmacology, neurobiology, all that thing is those are possible in the natural science course at Cambridge. You don't have to come in saying, yes, I'm going to be a chemist and I'm going to follow chemistry all the way through. You might start off being a chemist and then in the second year decide, no, I want to actually be a neurobiologist, and then in the end decide, no, I actually want to be a pharmacologist. And the options are there to move between these courses. But overall, we just try to give students a really, broad background so that they can appreciate all the different science subjects that actually feed in to whatever it is that you're interested in science.
I agree. Definitely, I have to say, I think it sets you up for multiple different types of careers as well. So I don't know if you know, but I was a student in the biochemistry department for my undergrad. And I mean, that set me up as a science communicator. So you don't have to stay in research based on the Cambridge Natural Sciences course as well.
No, I think it's only about...
now from...
Oh. Yeah, carry on.
Sorry, go on.
I was going to say only about 50% stay in research. So actually, there's a lot of people go off and do other things, and there's lots of people do fascinating things, like science communication, perhaps. Yes,
which is a very important field. We have a question from the public. So this person is curious about whether there are any cell signaling molecules that are known to not be ancient, given that most genes required to build neurons are also found in sponges.
Yeah, well, I don't know the answer to that, because people have only just started looking back, because it was always assumed that the nervous system was relatively modern. It evolved about the same time as animals, and certainly it was put together then. But as I say, the individual components that you see, like the molecules, like clearly the receptors that actually bind the neurotransmitters, were actually, it seems that they were around much earlier in evolution than we thought they were. And as I say, this is a very young subject, I should say. Oh dear, that's a bad pun. But really, people haven't explored how ancient how far these systems go back. But my feeling is that quite a lot of them will go quite a long way back in time.
That's very interesting. There don't appear to be any other questions in the chat. I don't want to put Latika on the spot, but I don't know if Latika has anything that you want to ask.
Actually, going with the nitric oxide route. So you say it helps relax smooth muscle. Would it be useful, therefore, like if you were at the gym, could it help in terms of like, I don't know, pumping iron and just getting it all done really quickly?
So you mean you just cheat, that you actually just turn on your nitric oxide and just stand there and well, actually, your muscles relaxing though, rather than tensing. And what you, so I don't think that's quite that.
I suppose would it lead to not having as sore muscles as you would normally?
I don't think so. No, sadly, I don't think that's going to be very useful. You're going to have to stick with the sore muscles for a bit.
Okay.
It would be lovely if there was a way to choose the gym. Yeah. I'm not a huge fan, so if there was a tablet that you could take with nitric oxide that would do it for me, that would make it so much more appealing. It doesn't look like there's any other questions, and we are 1 minute over our scheduled time anyway, so I think we'll wrap up this session. So I just want to say thank you very much, Sarah, for opening our series of Cambridge Festival talks with such a fascinating session on cell signaling. That was really interesting, so thank you very much.
You're welcome.
Thank you also, Latika, for helping run the event behind the scenes. Much appreciated.
No problem.
And then I just want to say also, and then I also just want to say thank you very much to all of you for joining us this afternoon and for watching. So please stay safe, and hopefully we'll see you again soon for one of our other Cambridge Festival talks. So thank you very much.